All right, back for one more. This is the technical part of the talk. Now there's just a few of us around, I want to say thank you for listening this far. Here's the data, and I don't want you to ever take anything what I say. But I also realize that these papers are really hard to read, even for me, who's been in the field for a while, and so I want to try to translate them for you. Here's my quick part of translation, with the idea that you can pause on this, you can go to the paper, you can go back and forth, and feel free to ask me questions as we go through this. Where are these 4 studies that I showed you, showing that T cells may learn to recognize COVID-19? And then after that, I'm going to talk a little bit about what is this project exactly that I gave my students to investigate similarities between common cold and SARS-CoV-2. So if you want me to give more details, I thought you'd never ask. I was looking for a Monty Python one of these, but I couldn't find one of them, which means our memeing ability as a nation, I believe it's in trouble. But Willy Wonka will do. Paper number 1. This is the one that came out way back in April. This is 34% of people from uninfected, they're seronegative. They respond to epitopes from the SARS-CoV-2 spike protein. And that's just one protein, like 10 to 20% of the genome. Here's the evidence that the proteins are similar. On the left, they have a bar chart of the different cold viruses and how similar they are to SARS-CoV-2. The higher the bar, the more similar they are. And you can see all 4 cold viruses, they look 25 to 40% similar. This is just for one protein, the spike protein, and the virus has dozens of proteins, like 2 dozen proteins when you really add it all up, depending on how you define protein, right? So the thing is, just looking at the spike protein, it looks awfully similar. It's also going to be a target for the immune system, so it's a good place to start. I want to show you the 2 graphs, D and E, over there. You can see that there's reactive healthy donors compared to COVID-19 patients in part D. The dots are higher in COVID-19 patients, but the dots are up there with reactive healthy donors. There is some portion of the T cells that will react. You get less of a reaction, but of course you do. The immune system is not activated against the virus until you're actually infected with it. The important thing is, do you have memory? Do you have any sort of shield? And now that we know you have a shield, let's figure out how big it is and how effective it is. If you look at the bars down in part E, you can see overall what percentage of donors have T cells that will actually react. And you can see that it's about 10 to 30% of healthy donors that have T cells that will react to this one protein. And that adds up to be 34% overall. That's still good news. Just one protein, and we have 1/3 of the people have T cells against it who have never been infected. So Grifoni et al. This is one of my favorite papers of this year. And this has been published in Cell. I've known it since it was born. So since it was publicly posted, and it has the big number that 50% of people have one kind of T cell, 20% of people have another kind of T cell, and it comes out to be more than 50% of people have one of the 2 kinds of T cells against SARS-CoV-2, even though these people, the sample was collected before SARS-CoV-2 ever made the jump from bat or pangolin into humans. This is the graphical abstract from this paper, and I really want to, if you want to look at a paper, look at this one, look at the graphical abstract. It's got a really nice one. If somebody's infected with COVID-19 on the top up there, you can see that they have 100% of those people who were infected has CD4 T cells, 70% had CD8 T cells. And you see that they have these S protein, M protein, N protein. If you've watched the structural immunology lectures, you know those are the 3 main structural proteins, and there's one more that they don't show, that are part of the virus in every part of the virus. Then you have the NSPs, which are going to be all the rest of the protein that works inside the rest of the virus that works inside the cell. But those are made by virus-infected cells. Those can be used to recognize the virus. And so you can see that you have 100% of people have antibodies, IgG antibodies. 80% of people have IgA antibodies who've been infected. 100% of people have CD4 T cells, 70% have CD8 T cells. Now move to the bottom, and you see how they have a question mark, but they say common cold coronaviruses, and they have 4 of them because there are 4 major strains. That's a good number. Somehow, we think it's from this, something has infected unexposed people. that they have CD4 T cells. 50% of them have CD4 T cells against the S protein, the M protein, and some of the NSPs. The important thing about this, it's not all of the NSPs, and it's not against the N protein. Notice this. So we can say, okay, does this make sense? If we look at the cold viruses, do we see a similar pattern of proteins being recognized by the immune system? And what my students found out is that yes, we do, down to the original peptide, that we can see the similarities among them. This, for example, is one of my student slides that brought up about the way that this is substantial response from the T cells, and you have CD4 T cells and CD8 T cells on this, and you can see unexposed. The numbers are not as high for the unexposed dots on all these graphs, but they're there. You know, the numbers are there, and they are brought up when infection happens. And so you have measurable T cell responses to the virus, even if you have never seen it before. Here's paper number 3. I've talked about this in detail in the structural immunology lectures, so I'm just going to mention it quickly now and refer you back to those. But this one found that there were 5 different spike protein. Again, it only looked at the spike protein, and it used kind of an unusual system. It's kind of an artificial system, but it's molecularly defined, and so we can do things like say, this exact peptide caused this exact T cell receptor to activate. And that's really cool. As a biochemist, I really like seeing that. I also know people who are on this paper, so I'm just very happy to see that what we can do as an institution, my college, produced students who worked on this. So this is the cool system, the cool biochemical system. It's called single chain trimer libraries. The single chain trimers are the sort of blue and green things in the middle. If you look at, this is kind of busy, but the non-circular things up on top, if you look way up there, those are single chain trimers. They're MHC proteins, and again, I talk about them more in other parts of it. But I want to say that you get down to getting to know exactly which little peptide, the colored peptide that's in the blue protein up there, you get to see exactly which peptide is causing the response, and you can even sequence the T cell receptor, and you can see exactly which part of the T cell receptor is interacting with that peptide, MHC complex. And so you see above my head, you can see CDR3 alpha and CDR3 beta. These are the exact sequences of the T cell receptors that are causing this reaction. So this is a little bit of an artificial system. It's a little bit focused on the spike protein, but like I said, 4 out of the 5 common epitopes that they found were predicted by just saying these 2 common cold strains look a lot like SARS in this particular location. This is how they cut up the spike protein. So this is the whole spike protein, and they were only tested a subset of the spike protein. You can only do so much. They made like 100 different single chain trimers, and overall about 1/3 of them actually worked well enough to get results from. So we've got to realize that if we didn't get results from certain things, it's possible that it works in a natural system. It just didn't work in their artificial system that they have here. So there's a trade-off between the detail that you get molecular biology level and the response that you get from the ability to say this is what happens naturally, the ability to look at the entire protein. But you can see that the colored bars, the little colored bars, are scattered throughout the protein, and those are the ones that worked, that they actually got through their system, and they were actually able to test them against T cells. Actually, all of these 100, they tried to test them, but they didn't all make it all the way through the system. So yes, I believe that the bolded/colored little bits were the fraction of the protein that they literally tested against T cells. Anyways, you can look at that more if you have the inclination. The other thing is that this is a really cool one because it has a lot of people involved in it. It has about 200 people on both sides of the study, 200 exposed individuals, 200 unexposed. And the thing is, they found pre-existing T cell responses in 81% of the 185 unexposed individuals. That's a lot of data, and that's a lot of evidence that this is real and this is going on. That's a lot of people to have this. Now, you know, a lot of them are probably so weak that they're not really effectual, but 81% of people walking around with some sort of immunity to SARS-CoV-2 anyways, I want to shout this from the rooftops because I think people need to know. we will get through this. It's been hard, and it's going to continue to be hard, especially for people who are living in outbreak states. I'm just hoping that, again, I keep hoping for some good surprises, and the good surprise I can say that's happened so far, I haven't seen it come back in full-fledged to where it has been a true big outbreak before. So it looks like this disease does not come back as much as we were afraid of. This T-cell immunity might be one of the reasons. Here's the data that I want to show you. They actually looked at which peptides were cross-reactive. Now, they didn't get down to the individual peptide level. They made a pool of all these cross-reactive ones. The other thing that they did is notice that they have HLA restriction there. Our particular studies was only on one HLA. It was the A*02 HLA. And notice that that's not even in the cross-reactive pool for that. But even so, the two of those peptides were peptides that we found with our study. So with all the strikes against it, we still found that like 2 out of the 7 possible peptides that we could have found with cross-reactivity actually showed up. I think that's significant considering all the ways that this could have gone wrong. So the actual data, they actually took that peptide and it activated T cells from non-infected patients. The bottom peptide affected T cells from a non-infected patient as well. And so we know that this peptide activates T cells in non-infected patients. We know that this peptide is found, essentially the same peptide is found in cold coronaviruses as is found in SARS. And so that connects all the dots. The only question is, how many people, what proportion of people is this effective in? And so I want to go ahead and I want to go really fast through all this because someday I'm going to go through in more detail, opening up this project to how other people can participate. But I want to say real quickly, how did my Biochemistry III students actually help do this, find the places where the common cold and SARS-CoV-2 are similar? Remember how I said that you can look at the letters of each gene. And if you look at the letters, if you compare the letters, if the letters are the same, then it's going to look the same. If the letters look the same to your eye, the letters are going to look the same to the immune system and to all the other things. So, how did my students look on this? I thought you'd never ask. Here is the structure of the entire protein, and I just want to point out that we looked at the green proteins and the orange proteins on this. We did not look at the gray or gold proteins because I could only give my students so much work. And also there was indication that they didn't provoke as much of an immune response either. I basically looked at what we did. The important thing is that we looked at way more than just the S protein. There might be some immunity hiding out in the other parts as well, but we definitely at least caught most of the immunity. And here's a nice little tree that shows you the relatedness. This is like the family tree of viruses. And if you notice, there's 2019 nCoV is right there. It's right above SARS-CoV, which we would expect. Those are very closely related. But if you look at the squares, those are the 2 cold strains that we did. One of them is a beta coronavirus, so it's in the same branch of the family tree as 29 nCoV, SARS-CoV-2. And you see that 229E is an alpha coronavirus at the very top there. The HKU1 below that, and let me find the other one, NL63 at the very top. Those are the other 2 of the 4 cold coronaviruses. But again, I could only give my students so much work, but I did want to make sure that we looked at more than one cold coronavirus, so we looked at 1 beta and 1 alpha. By the way, we found the relatedness that you would expect. The OC43 is more related to the SARS-CoV-2 than the 229E is. But they're both related. They're both fairly related. They both have a definite family resemblance. Here's where we actually went through each of the proteins and I compared the percent identity. You can see that the open reading frame, we're talking about a 30 to 40% identity. People who know about proteins and compare proteins know that like 20 to 25 is like the floor. So this is above the floor. It's not above 50%, but what you need is you need some short stretches of the protein to be more than 50% similar, not the whole protein. And within a protein that's 40% similar, you're going to have some stretches that are 50% similar. That's the whole idea. And those stretches can be recognized by the same T cells that recognize the virus. So here's all the data that I did. I put it up. You can pause it if you want to look at it. And what they did, and I can give the protocols, I'm going to put this online eventually. what you do is you take the 3 different genomes and you express them as amino acids on your computer, and then you stack them up. And you can see, yeah, they don't match exactly. On the left, they're not matching very much. But on the right, you can see there are some places where they have the same colors or even the same letters. This means they are chemically similar or identical if they have the same letter. And so you can just do this, and you can say on the C-terminal part, on the right-hand part of the screen, you have more similarity than on the left-hand part. How much similarity? Well, you want to have more than, say, 50 to 60% similarity to have a good chance of tricking the immune system into thinking that you're seeing the same thing. The good news is, there are several places that we know will trigger the immune system. We predict will trigger the immune system. And a lot of those places, like I said, there's like 100 possibilities for cross-reactivity between these 3 viruses. So what they do is they go through a whole series where you find and copy the corresponding sequences. You use this program called CView. You find that place. So this is what a student, and the student who did this, by the way, is named Tristan. So thank you, Tristan, for doing this. You have... You can see that you have the same letter. You find it in the file that has all of the different epitopes in it. And you compare it to the OC43 sequence, so the OC43 and the SARS sequence here. You can see that they are not identical, but they're pretty similar. There's a lot of letters that are the same. So you note where they start and stop. You enter that onto the data sheet, and you say, I have found a similarity. You look at that right there. You count how many residues are identical, and you put that in. And you see right here, you calculate the percentage from that. So that's what you do on the computer to find out where the similarities are. You're using your eyes to do what the immune system does. And again, you have here, and here you have some high degrees of similarity for short little snippets that the immune system recognizes. The white boxes here, you can see with your eye how similar they are. And you can see if the white box, if you have a T cell that interacts with the white box letters for 229E up top, and then it sees SARS-2, the only difference is that the S has been turned to an E. It's very likely that that T cell will be able to interact with SARS-2, even though it's never seen SARS-2 before. That's what we're talking about. And so the students went through the entire genome that we have. This is the genome parts that we looked at in order. Where it's red, they're not similar. Where it's green, they're more similar. And you see there's a lot of green here. And each of the students was given a different part of the genome to look at and to say, how similar are these? You notice there are 2 bars. One bar is for the one cold strain, and the lower bar is for the other cold strain. But you can see that the patterns are slightly different, but there still is a tendency, and there's a tendency to have more green on the right side. And so here's where I found all the little snippets that look the same to us and probably look the same to the immune system. There's a lot of boxes here. There's a lot of similarity. The students who happened to be assigned the right side of the sequences got a lot more stuff than the people on the left-hand side of the sequences. And that's just the alphabetical curse, I guess. Here's where we divided up the non-structural proteins, and the ORF1 is divided up into several non-structural proteins. The important thing about this is we compared this to Grifoni et al., and we wanted to say how many epitopes did we find in each non-structural protein. If you look at this, the non-structural proteins on the bottom of the screen have bigger numbers, more epitopes, more recognizable. This goes along with what we saw on the last one because they're on the right-hand side of the screen, they're on the lower part of the screen, they have more epitopes that are there. And so if you put a red box around all of these ones that had a larger number of epitopes that we found a lot of cross-reactivity among, lo and behold, those tend to go, with the exception of the purple box, like I said before, those tend to go along with what Grifoni et al. Found for the uneducated immune systems somehow responding to this. Where we found no cross-reactivity, they saw lower reactions. And so just a couple of things that my students made. This is showing the spike protein and showing where the little snippets are that interact with the immune system. This is Suzanne Stafford made this picture of, they found that the OC43, they were, these are some snippets that are the same for OC43 and for SARS, the green parts. It's only a little bit of the protein, but that's all it takes for the immune system to be activated. Here's an example of cross-reactivity found between both cold strains and the sequence itself. You can see that those letters are pretty much the same. Being in the second half of the alphabet for the way that we did this, this is Precious Nin, and she found a lot of similarity. She had a lot of boxes on that previous graph. Here's from Borith King, and he found a lot of similar sequences as well. You can see there's a lot of those sequences, the letters are the same, and we can go and we can even find places in the literature where those letters activate T cells. Caroline Kim made this. She found a lot of letters the same as well. And this is Lam True. Lam found a lot of letters the same as well. As we move on through the alphabet, you see how we're getting more and more of this. Again, that's just the way that I set this up. But you can see that there's a lot of letters that are the same. When the letters are the same, the immune system can react to it. And finally, here's Tristan again. Tristan gave us some pie charts. There's a lot of similarities above 30%. That's a little bit lenient. I think that looking at the literature, we need similarities 50 to 60% to be able to say that cross-reactivity is a good chance. We also need affinities that are below 500, and so you can see that there's a lot of those, is what Tristan's showing here. So thank you to all the students for making these. Thank you to you for listening. Let me know what questions you have, and I hope someday to be able to have this set up online so that you can do the exact same thing.